Hi, my name is Julian Stamp. I'm a PhD candidate at the Center for Computational Molecular Biology at Brown University. In this talk, I'm going to present to you the method I just published, the multivariate marginal epistasis test, which is a story about how to leverage genetic correlations between traits in genome-wide association studies of epistasis. I will start with a brief introduction to the problem I'm addressing. I will explain the method and then show in simulations and on real data what this method can do before concluding with a brief recap. A key motivation of genetic research has been to understand the link between genotype and phenotype. In 2003, the Human Genome Project was completed, and hopes were high for a full understanding of human genetics and its effects on human traits. The idea was to genotype large cohorts of individuals and through simple statistical tests create mappings between genes and human traits. 20 years later, millions of individuals have been genotyped and thousands of genome-wide association studies have been performed, and we've come to realize that things are not that simple. Some of the open problems in research about complex traits are, for example, when rare variants, why is it moving by itself? When rare variants contribute to trait variation, poor accuracy of predictions of phenotypes, and how to explain the gap between the known heritability of traits and the heritability accounted for by discovered variants. We think of phenotypic traits as being affected both by genetics and environmental factors. The amount of variation that is explained by genetic factors we refer to as heritability. We can further partition heritability into additive and nonlinear components. The method I want to talk about draws from this picture of trait variance partitioning to make inferences about epistasis. If we have all phenotype and genotype information possible, that is combinatorially complete data, we can simply study the trait architecture with a linear transformation, the Hadamard-Walsh transform. In this transformation, psi is the Hadamard matrix, W the vector of trait values, and sorry, does anybody know how to fix the auto play?
I'm just going to exit the presentation.
Okay, I'll try again. So in this transformation, psi is the Hadamard matrix, W the vector of trait values, and E is the vector of epistatic effects. Each row in this transformation corresponds to a genotype with an associated trait value. The Hadamard-Walsh transform is a linear transformation between the trait domain and the epistatic effect domain. What we can measure are the trait values, but what we care to know is the effect size domain because it contains the full information of the trait architecture. In most cases, we have limited access to combinatorially complete data. Biobank scale data typically has on the order of 100,000 to 1 million SNPs and 10,000 to 100,000 samples. With a minor allele counting encoding, already for 170 SNPs, there are more combinations thinkable than atoms in the universe. So in general, we need to work with underdetermined and combinatorially incomplete data. The problem of studying complex traits is a challenge. There are more than 11 million SNPs in the human genome, and on the order of 400,000 trait associations have been reported. Through, for example, family and twin studies, we can get reasonable estimates of heritability for complex traits, but for most traits, the variance explained by associated SNPs is only a fraction of the known heritability. It is not clear where the missing heritability is. One idea is that either we have not found all SNPs because there are many, many SNPs with small effects that are hard to find with GWAS, or that there are rare variants with large effects, but those are also hard to find with GWAS. Another explanation could be epistasis. Interactions, even of known variants, easily could account for a large portion of trait variants. However, computational methods to detect epistasis are mostly either underpowered or resource intensive. One problem why many methods are underpowered is the search space. Even if we only look at pairwise epistasis, the number of tests required scales quadratically already. But there's a solution to this particular problem. The idea is to marginalize over the pairwise interactions and reduce the search space to something more manageable. And it works roughly like this. Take this cartoon of pairwise interaction effects plotted as a heat map, and the color and dot size encodes effect size. Larger, darker dots are larger effect sizes. In an explicit search, where we test each interaction individually, we would have to test this upper triangular space, and the number of tests scales quadratically. The quadratic scaling of number of tests leads to severe loss of power after multiple testing correction. The idea of marginal epistasis now is that we can marginalize over all interactions for a given SNP. For example, I can marginalize over all interactions of SNP K with all other SNPs and accumulate the evidence of many interactions into stronger effects that are easier to detect. One, because they're larger, and second, because now we only correct for testing each SNP once instead of many times. Genetic correlations between traits may cause correlated response to selection. Both pleiotropy and linkage disequilibrium create genetic correlations between traits so that traits do not vary independently of one another. Some assume pleiotropy and perfect linkage to be identical. Others, however, argue that this is wrong and that strong genetic correlation between traits is most of the time maintained by pleiotropic variants rather than tight linkage, at least in the range of low mutation rates of the sorts we find in mammals. For GWAS, looking for linear effects, it has already been shown that multivariate models can improve detection of causal variants. So I set out to leverage genetic correlations between traits in genome-wide association studies of epistasis. My advisor, Lauren Crawford, published the method of the marginal epistasis test. It is based on a linear model. What we see here is a model that models a quantitative trait, y, and what goes into this trait is a population mean, linear contributions of variance, and environmental effects. The part of the model that we really care about are the interaction effects. This model explicitly models pairwise interactions and partitions the heritable portion of the trait variance into an additive and a pairwise epistatic variance component. By assuming random normal effects, these variance components follow multivariate normal distributions. The additive variance component covariance is the genetic relatedness matrix, which accounts for the polygenic background and population structure. The important part of the model comes from the capital G matrix, which captures the interactions of SNP K with its background. Statistical inference is performed using a variance component estimation method to jointly estimate these model parameters. For MAPIT, we only consider one trait, only covariance between samples, and we estimate only one variance component per SNP. For multivariate MAPIT, we extend this model to any number of traits, and something that changes in a significant way by doing so is that we not only have covariance between samples, but also covariance between the variance components of different traits. For D traits, we have to estimate D choose two variance and covariance components and combine the p-values in aggregate test statistics. In writing out this model as multivariate, however, I found that it models a very specific case. In this model, we have genotype data, the SNPs, denoted as X, traits denoted as Y. We have linear effects, and in general, one SNP could have interactions with different SNPs affecting different traits. Even if these effects, the alpha, were correlated, multivariate method does not pick up on this structure in the multivariate analysis. The way the math works out, multivariate method models explicitly pairwise interactions having correlated effects on multiple traits. One interaction affects two traits and the effects are correlated. Multivariate method models the right case and gains sensitivity and power when the effects of one interaction on multiple traits are highly correlated. We can show in simulations that the method works. What we care about in simulations is simulations under the null hypothesis to show that we can control type 1 errors, and under the alternative hypothesis to show power and sensitivity. To simulate data, we need to control the broad sense heritability and the proportion of variance due to epistasis, which in this slide I parameterize with rho. To simulate pairwise interactions given genotype data, I selected causal SNPs at random, some of which purely have additive effects, here yellow, and the ones that also have epistatic effects were randomly partitioned into two groups. Each SNP in one group is simulated to interact with each SNP of the other group, here shown as edges. For each edge, epistatic effects are drawn from a multivariate normal distribution to match the model assumptions. With all this, I then compute traits that have known genetic architecture. To show how this translates into marginal epistasis, here are two examples. The trait portion that is due to epistasis involving SNP 1 is GX1, and it involves interactions with SNPs X3, X4, and X5, and the same for SNP X3. Here, it only involves interactions with X1 and 2. This also illustrates how this kind of modeling accumulates contributions of multiple interactions. In terms of the pie charts, the share of variance explained by SNPs from a group with less members is larger. What we try to detect with multivariate method is exactly that, the portion of trait variance due to all interactions of one SNP with many others. Therefore, we expect it to be easier to detect SNPs from group 1 here compared to group 2. The method has good error control. QQ plots show whether the p-values of the method under the null hypothesis follow the expectation under the null. Note that the QQ plot for the covariance component, where two traits are analyzed simultaneously, is actually deflated, making the p-values of the covariance statistic rather conservative. What you see here are empirical power estimates based on simulations. I'm evaluating the power at significance threshold 0.05, and I simulate two traits with correlated effects but different heritability. For one, 60% of trait variance is heritable, for the other, only 30%. V12 is the strength of the correlations between the epistatic effects on traits 1 and 2. This figure shows that the power of the covariance statistic on its own increases when the correlation between the effects increases, and that the combined statistic improves the power of the method over the univariate case. The ROC curves show something interesting too. In general, ROC curves tell you whether a method correctly prioritizes true positives. The figure shows you data from simulations with heritability 0.6 and high correlations between the epistatic effect sizes on the two traits. Steeper curves indicate higher sensitivity. The dashed lines are based on the trait-specific variance component estimates and correspond to the benchmark of the univariate case. The purple line is based on the p-value from the covariance component estimates. The pink line is combining both univariate statistics and the covariance statistic. With that, we see that when the trait variance due to nonlinear effects is low and the correlation between the effects is high, the sensitivity of the covariance statistic, the purple line, suddenly becomes very sensitive. This is important to interpret the results I got from applying this method to hematology traits in mice. The traits you see here are in the left column hematocrit and hemoglobin, and in the right column mean corpuscular volume and mean corpuscular hemoglobin. What you see is that the variance component of both traits individually, the first two rows, which correspond to a univariate method, do not show any significance, whereas the covariance components show significant SNPs. In multivariate method, we then summarize these three analyses into one combined p-value. Both of these traits are highly correlated, or pairs of traits are highly correlated. The variance estimate corresponding to the first two rows is the special case when both traits are the same, that we find a strong signal in the covariance statistic and not in the variance statistic when the traits are highly correlated shows that the model really picks up on the signal of high correlation between epistatic interactions that affect both traits. That the signal of the covariance picks up the covariance picks up indicates highly correlated effects, but low overall heritability, as the ROC curves illustrated. Interestingly, this pattern of only the covariance showing a strong signal shows up in most traits that I analyzed. What you see here is a facet plot, where each facet corresponds to a pair of traits analyzed with multivariate method. On the diagonal, you can see the univariate case of only one trait. As you can see, there are very few plots on the diagonal that show any signal. On the off diagonals, you see the combined Manhattan plots of the multivariate analysis. This pattern is independent of the correlation of traits. It indicates signals that are coming from traits with high genetic correlations of pleiotropic effects of pairwise interactions. To conclude, I want to highlight the strengths of this method. Multivariate method is a tool for the analysis of shared genetic architecture of traits. As linear mixed model, it controls for the confounding due to population structure. It leverages correlations between effects, and if present, it gains a lot of sensitivity. By marginalizing over all pairwise interactions of a given variant with any other variant, it accumulates weak effects into a strong signal. And a second effect of the marginalization is that it reduces the search space and by that avoids the problem of a large multiple testing burden. I want to thank my advisors, Lauren Crawford and Daniel Weinrich, for the guidance and support throughout this project, and the Crawford Lab members and Dave for the many discussions and constructive criticism in pursuing this work. Lastly, I would like to point out that the multivariate mapit software is published as R package with documentation on GitHub pages, and it is also available on CRAN so that users of R can simply install it in the R console. Thank you.
Thank you so much, Julian. There's time for 1 or 2 questions. If you have questions, please come down. We'll see if anyone comes down, but I had a question for you. Yeah, thanks for the nice presentation. I was wondering, how does the scalability work? I noticed that you applied your data to mouse genomes, and maybe you can comment on how scalable it can be.
Yeah, this is probably the weakest point of the method because it's a linear mixed model. It has a major computational overhead, and I haven't found a way to speed up the matrix math that is required to make this method work. So in terms of sample size, it scales in best case quadratically, probably a little worse.
All right, so more